Part 1. The Lipid Foundation of Human Structure. Chapter 1. The Body as a Lipid System. The human body is organized around boundaries. Every function, from cellular metabolism to whole system coordination, depends on the ability to maintain separation between internal and external environments while allowing controlled exchange. This separation is not conceptual. It is built from lipids. Each cell is enclosed by a membrane composed primarily of phospholipids, cholesterol, and embedded proteins, forming a dynamic structure that defines what enters, what exits, and how signals are transmitted. Without this boundary, gradients collapse, signaling fails, and the distinction between inside and outside disappears. The body does not begin with energy. It begins with containment. Membranes do more than enclose. Their composition determines how the system behaves under load. The arrangement of lipid tails affects fluidity, which in turn controls how proteins embedded in the membrane can move, interact, and function. Cholesterol modulates this environment by stabilizing the membrane, preventing excessive rigidity at low temperatures and excessive permeability at high temperatures. These properties are not static. They shift continuously based on the materials available to the system. When lipid composition changes, the behavior of the membrane changes with it. Permeability alters. Electrical resistance shifts. The ability to hold ion gradients either strengthens or weakens. These changes propagate outward, affecting every process that depends on stable separation. Electrical stability emerges directly from membrane integrity. Cells maintain differences in ion concentration across their membranes, creating gradients that store potential energy. This stored potential drives nerve impulses, muscle contraction, and transport processes throughout the body. If the membrane becomes too permeable, ions leak across it, forcing the cell to expend energy to restore balance. This constant correction increases baseline demand while reducing efficiency. The system begins to operate under strain, not because energy is lacking, but because structure no longer holds the conditions required for efficient use. Signal transmission depends on organized lipid environments. Receptors are not randomly distributed across the membrane. They cluster within specific regions where lipid composition supports their function. These microdomains allow signals to be initiated, amplified, and terminated with precision. When membrane composition becomes unstable, these domains lose coherence. Receptors drift, clustering becomes inconsistent, and signaling pathways activate without proper coordination. The result is not simply reduced signaling, but distorted signaling, where messages are initiated, prolonged, or terminated incorrectly. The system receives input, but the interpretation of that input becomes unreliable. The body extends this lipid architecture across multiple scales. Organelles within the cell are also defined by membranes, each with distinct compositions tailored to their function. The mitochondrion relies on a tightly controlled inner membrane to maintain the gradients required for energy production. The endoplasmic reticulum uses its membrane structure to synthesize and process lipids and proteins. Vesicles transport materials by merging and separating membranes in controlled sequences. At the tissue level, cells coordinate through membrane interactions, forming barriers, junctions, and communication networks that maintain system-wide organization. This continuity reveals a single principle repeated across scales. Function follows from how boundaries are constructed and maintained. Dietary lipids feed directly into this architecture. They are absorbed, transported, and incorporated into membranes through ongoing remodeling processes. This integration is not selective in a way that filters out all variability. The materials available influence what is built. If the incoming lipids are stable and resistant to degradation, membranes move toward greater coherence. If they are prone to oxidation or structural instability, those characteristics become part of the system. Over time, this alters how cells behave, how signals are processed, and how energy is managed. The body is not insulated from its inputs. It is continuously rebuilt from them. Viewing the body as a lipid system resolves inconsistencies that arise under energy-based models. It explains why identical caloric intake can produce different outcomes, why signaling becomes unreliable under certain dietary conditions, and why restoring stability often requires changes in material input rather than adjustments in energy balance. The system is not reacting unpredictably. It is expressing the properties of the structure it is built from. When that structure is stable, behavior becomes consistent. When it is not, variability emerges as a direct consequence of degraded boundaries. This framework establishes the foundation for understanding physiology as a function of material composition. Lipids are not a secondary component of the system. They are the medium through which the system exists and operates. Every subsequent process, from signaling to energy production, is constrained by the properties of this medium. To understand how the body functions, it must first be understood as a system built from lipids, where boundaries define behavior and structure determines outcome. The body as a boundary-defined system. The body operates by maintaining separation. Every function depends on the ability to establish and preserve differences between compartments, from the level of a single cell to the coordination of entire organs. This separation is enforced by physical boundaries built from lipids. Each boundary defines what is contained, what is excluded, and how exchange is controlled. Without these boundaries, gradients collapse, concentrations equalize, and the conditions required for organized activity disappear. The system does not begin with reactions. It begins with the ability to hold distinct internal states. Cell membranes form the primary layer of this organization. They create a controlled interface where transport is regulated rather than allowed to occur freely. Channels, transporters, and receptors operate within this boundary, but their function depends on the stability of the structure that holds them in place. If the membrane becomes too permeable, ions move across it without control, forcing the cell to expend energy to restore separation. If it becomes too rigid, necessary exchanges slow or fail. The boundary must hold a narrow range of physical properties to maintain function. These properties are determined by the lipids that compose it, not by the energy moving through it. This principle extends inward. Organelles maintain their own boundaries, each with specific compositions that define their role. The nucleus isolates genetic material while allowing selective transport of regulatory molecules. The mitochondrion maintains a steep gradient across its inner membrane, enabling controlled energy conversion. The endoplasmic reticulum forms a network where synthesis and processing occur within a defined environment. Each of these compartments depends on its boundary to maintain the conditions required for its function. When those boundaries degrade, the processes they support lose precision or fail entirely. Gradients are the operational currency of the system, and gradients only exist when boundaries hold. Differences in ion concentration, electrical charge, and chemical composition drive movement and enable work to be performed. When separation is maintained, gradients can be established and used efficiently. When separation weakens, gradients dissipate and the system shifts into constant correction. Energy is diverted toward maintaining basic differences rather than performing higher-level functions. This increases demand without improving output, creating a state where the system is active but inefficient. At larger scales, tissues and organs rely on coordinated boundaries to function as integrated systems. Epithelial layers control what enters and exits the body. Vascular endothelium regulates exchange between blood and tissue. Neural networks depend on membrane stability to transmit signals with speed and accuracy. These structures are not independent. They operate as a continuous boundary system where local instability can propagate into systemic effects. A disruption at the cellular level can scale into tissue dysfunction because the same structural principles apply across levels. The body does not manage complexity by increasing reaction rates or energy throughput. It manages complexity by maintaining organized separation. Boundaries allow different processes to occur simultaneously without interference. They create controlled environments where specific reactions can proceed under defined conditions. When this organization is intact, the system operates with clarity. When it breaks down, processes overlap, signals interfere, and the distinction between compartments begins to blur. Understanding the body as a boundary-defined system reframes how inputs are evaluated. Materials that reinforce boundary integrity support the entire system. Materials that weaken boundaries introduce instability that propagates across functions. This is not an abstract effect. It is a direct consequence of how physical structures behave under load. The body maintains order by maintaining separation, and that separation is built from lipids that determine whether the system holds or begins to lose coherence. Lipids as the primary structural material. The body is constructed from materials that determine how it holds shape, maintains separation, and supports function under load. Lipids form the core of that material system. Every cell, organelle, and transport interface depends on lipid assemblies to create boundaries that are flexible enough to allow controlled movement yet stable enough to preserve internal conditions. These assemblies are not inert layers. Their composition sets the physical properties of the system, including thickness, packing density, permeability, and electrical resistance. When lipid composition shifts, these properties shift in parallel, altering how the system behaves at every level. Phospholipids provide the foundational framework. Their amphipathic structure drives self-assembly into bilayers, orienting hydrophilic heads outward and hydrophobic tails inward to create a continuous barrier. This arrangement determines baseline permeability. Water and small nonpolar molecules pass differently than ions and polar compounds, and the membrane enforces that distinction without constant energy input. Cholesterol intercalates within this framework and regulates packing. It limits excessive movement when membranes become too fluid and prevents tight packing when they become too rigid, maintaining a narrow operating range where transporters and receptors can function consistently. The ratio of saturated to unsaturated fatty acids further tunes this environment. Saturated chains pack tightly and resist deformation. Unsaturated chains introduce kinks that increase spacing and mobility. The balance between them determines whether the membrane holds structure under stress or drifts into instability. These material properties control protein behavior. Membrane proteins do not function independently of their surroundings. Their orientation, movement, and interaction rates depend on the lipid environment that supports them. Receptors require specific packing conditions to cluster and initiate signaling. Transporters rely on membrane thickness and fluidity to cycle between conformations. Enzymes embedded within membranes depend on local composition to maintain catalytic efficiency. When lipid structure becomes inconsistent, protein function becomes inconsistent even when protein levels remain unchanged. The system does not fail from a lack of components. It fails because the environment no longer supports reliable operation. Electrical behavior is set by lipid structure as well. Ion gradients are held across membranes because the lipid bilayer resists free ion movement. This resistance allows cells to store potential and release it in controlled ways. If the membrane becomes more permeable due to altered lipid composition or oxidative damage, ions leak and gradients weaken. The cell compensates by increasing active transport, raising energy demand without improving output. Over time, this shifts the system into a state where energy is consumed maintaining separation rather than performing work. The difference between efficient and inefficient systems is not how much energy they receive, but how well their structure preserves the conditions required to use that energy. Lipid material also determines durability under chemical stress. Double bonds within unsaturated fatty acids introduce points of vulnerability where oxidation can occur. When these sites are attacked, lipid chains break or form reactive byproducts that disrupt membrane organization. This increases permeability and distorts local signaling environments. Saturated lipids lack these reactive sites and maintain structural integrity under the same conditions. The distribution of these materials within membranes therefore sets the threshold at which damage accumulates. Systems built with more stable lipids resist disruption. Systems built with more fragile lipids require constant repair and operate closer to failure. The body maintains this structure through continuous remodeling. Lipids are not fixed once incorporated. They are replaced over time as new materials enter through diet and existing components are removed or repaired. This process integrates incoming lipids directly into the architecture of the system. The properties of membranes gradually shift to reflect the available material pool. If that pool favors stability, membranes become more coherent and resistant to disruption. If it favors instability, those characteristics accumulate within the system. The outcome is not immediate, but it is consistent because the structure is being rebuilt continuously from the same inputs. Recognizing lipids as the primary structural material clarifies why changes in diet produce systemic effects that extend beyond energy balance. Lipids do not simply pass through the body. They become the body. They define the environment in which all biological processes occur. When the material is stable, processes align and operate with precision. When the material is unstable, variability emerges across systems that depend on consistent conditions. The behavior of the organism reflects the properties of the material it is built from, and those properties are determined by the lipids that form its structure. Separation, containment, and controlled exchange. Biological function depends on the ability to maintain differences. Every process requires distinct internal conditions that must be preserved against constant external pressure toward equilibrium. This is achieved through separation, enforced by lipid boundaries that hold compartments apart while permitting selective interaction. Containment is not passive. It is an active physical state where gradients are maintained, concentrations are controlled, and reactions occur within defined limits. Without this containment, chemical identity dissolves, gradients collapse, and the system loses the conditions required to produce organized output. Membranes establish this separation by creating a barrier that restricts free movement while embedding mechanisms for controlled exchange. Ions, metabolites, and signaling molecules do not move arbitrarily across this boundary. Their passage is mediated by channels, transporters, and carriers whose behavior depends on the properties of the lipid environment. The membrane sets the baseline resistance to movement, while proteins provide regulated pathways that open, close, and cycle in response to specific conditions. This arrangement allows the system to maintain internal stability while still interacting with its surroundings in a precise and directed way. Containment allows gradients to exist, and gradients drive function. Differences in ion concentration generate electrical potential. Differences in chemical composition create directional flow. These gradients are not static stores of energy. They are dynamic conditions that enable work to be performed when pathways are opened. If the boundary weakens, these gradients dissipate. Ions leak across membranes, concentrations equalize, and the system must expend energy to restore separation. This shifts the system from controlled operation into constant correction, increasing demand without increasing output. Controlled exchange ensures that movement across boundaries is purposeful rather than random. Nutrients are transported into cells according to need and availability. Waste products are removed in a regulated manner. Signals are transmitted when receptors engage and pathways activate. Each of these processes depends on the integrity of the boundary. If membrane composition alters permeability, transport becomes either excessive or insufficient. If receptor environments degrade, signals are initiated or terminated incorrectly. The system continues to exchange with its environment, but the exchange loses precision, leading to accumulation of error over time. This framework extends across all levels of organization. Cellular membranes maintain internal chemistry. Organelle membranes isolate specialized processes. Tissue barriers regulate interaction between compartments. The same principles apply at each level. Separation maintains identity, containment preserves conditions, and controlled exchange enables function. Disruption at any level propagates outward because each boundary is part of a larger system of coordinated separation. The materials that form these boundaries determine how effectively they perform these roles. Lipid composition sets permeability, resistance to stress, and susceptibility to damage. Stable structures maintain separation with minimal energy expenditure. Unstable structures require continuous correction and remain prone to failure under load. The difference is not conceptual. It is a direct consequence of how physical materials behave when subjected to chemical and electrical gradients. Understanding separation, containment, and controlled exchange as material properties rather than abstract concepts clarifies how the system maintains order. The body does not rely on constant intervention to keep processes aligned. It relies on boundaries that hold conditions steady and allow interaction only when it is required. When those boundaries are constructed from appropriate materials, function emerges with consistency. When they are not, variability increases because the system can no longer control how it exchanges with its environment. Why structure precedes all function? Function does not arise in open space. It requires defined conditions that determine what reactions can occur, how quickly they proceed, and how their outputs are contained. Structure establishes those conditions before any function can exist. Without a boundary to hold concentration differences, substrates diffuse, gradients collapse, and reaction pathways lose direction. The cell does not begin by performing work. It begins by creating a space in which work can be performed without interference. That space is constructed from lipid membranes that enforce separation and define the environment in which all subsequent activity takes place. Every functional process depends on constraints set by structure. Enzymatic reactions require specific concentrations of substrates and cofactors. Signal transduction depends on spatial organization, where receptors and downstream components are positioned within defined regions of the membrane. Energy production relies on the ability to maintain gradients across tightly controlled boundaries. These are not optional conditions that improve efficiency. They are prerequisites. When structure is intact, reactions proceed with predictable rates and outcomes. When structure degrades, those same reactions become inconsistent because the conditions required for their execution are no longer stable. Membranes determine how molecules move, and movement determines interaction. Transport is not governed solely by the presence of channels or carriers. It is governed by the physical properties of the membrane that surrounds them. Permeability sets the baseline for diffusion. Thickness and fluidity influence how transport proteins change shape and cycle between states. Electrical resistance determines how well ion gradients are preserved. These structural variables define the operating range of the system. If they shift, transport behavior shifts, altering substrate availability and reaction rates across the entire network. Signal generation and interpretation are equally dependent on structure. Receptors require organized lipid environments to cluster and initiate signaling cascades. Downstream components depend on proximity and orientation to propagate signals with precision. When membrane organization is lost, signaling becomes fragmented. Signals may initiate without proper amplification or persist beyond their intended duration due to delayed termination. The system continues to receive input, but the interpretation of that input becomes unreliable because the structural context that defines signal behavior has changed. Energy production provides a clear example of structure, setting the limits of function. The mitochondrial inner membrane maintains a proton gradient that drives ADP synthesis. This gradient exists only because the membrane restricts proton movement. If the membrane becomes permeable, protons leak, and the gradient weakens. The system compensates by increasing electron flow, which raises reactive byproduct formation and further degrades the structure. Energy continues to enter the system, but its conversion becomes inefficient because the structural conditions required for controlled conversion are compromised. Function does not fail from lack of input. It fails because structure no longer supports it. Time reinforces the precedence of structure. Structural changes accumulate through continuous remodeling, gradually altering the conditions under which all functions operate. As membranes incorporate different lipid compositions, their properties shift, changing how transport, signaling, and energy production behave. These changes occur before measurable functional outcomes appear. By the time dysfunction is detected, the underlying structure has already moved outside its optimal range. Addressing function without correcting structure leads to temporary adjustments that do not resolve the conditions producing the problem. Recognizing that structure precedes function aligns biological behavior with physical constraints. The system operates according to the properties of the materials that define it. When those materials support stable boundaries, functions align and produce consistent outcomes. When they do not, variability emerges across processes that depend on those boundaries. Function is not an independent layer that can be adjusted in isolation. It is the expression of the structure in which it occurs, and its reliability is determined by whether that structure can hold the conditions required for controlled operation.